Morning, everyone. My name is Jim Gardner. I'm one of the research scientists here at the Royal Tyrrell Museum. I'd like to welcome you to today's installment of our 2022 speaker series on this chilly, lightly snowing southern Alberta late winter day. Today, we're happy to host Dr. Daniel Paluh, who's a NSF postdoctoral fellow at the University of Florida and Harvard University. Dan was raised in Erie, Pennsylvania. He became interested in amphibians and reptiles while pursuing his Bachelor of Science degree at John Carroll University in Ohio, where he had the opportunity to work with several professors on projects involving turtles, salamanders, and poison frogs. After completing his undergrad degree in 2014, Dan went to Villanova University in Pennsylvania for a master's degree, which he completed in 2016. He worked on the comparative morphology and phylogenetic and functional implications of the quadrate bone in geckos, which are a fascinating group of lizards. Dan then headed south to the University of Florida in Gainesville, where for his PhD, he studied macroevolutionary patterns of phenotypic diversity across the frog tree of life, and he recently graduated late in 2021. Dan's research focuses broadly on the evolution of phenotypic diversity in amphibians and reptiles using museum specimens, micro-CT scanning, and phylogenetic comparative methods. So today, Dan will provide an overview of his PhD research in a talk entitled The Evolution of Hyperossification and Tooth Loss in Frogs, one of my favorite group of animals. So Dan, I'll turn it over to you and you can hop to it.
All right. Thank you for that introduction, Jim, and thank you for the invitation to speak here today. I'm very excited to be a participant in this museum speaker series to share some of my research on the evolution of frogs. So many biologists, myself included, are fascinated by tetrapods because of their amazing diversity in shapes and sizes. And this is especially true in some of the groups with the highest species diversity, including mammals, lizards and snakes, salamanders, and birds. So here we have the smallest and largest species in these groups for at least the living representatives, and we can see several examples of extreme body plan shifts, included elongated and truncated bodies, limbs transitioning to fins, to wings, to no limbs at all, and just like a really remarkable amount of diversity. But one tetrapod group that doesn't fit this pattern of having high phenotypic diversity with high species diversity is the frogs. And there are over 7,000 species of frogs described living today. So this is almost 1,000 more than all living mammals, but they have much less body size and body shape variation than these other groups. So here on the top, we have the smallest described species of living frog, Paedophryne from New Guinea, which is comfortably sitting on a dime. And this is actually also currently the smallest known vertebrate species overall. And then at the bottom, we have the largest known living species of frog, the Goliath frog from Central Africa, which is really big for a frog, but still smaller than a house cat. And the body plan for these two species is fundamentally the same. And this is also true for all of the other living species of frogs between these two extremes. And we can look to the fossil record to infer when this derived body plan evolved. The earliest known stem frog or proto-frog known as of today is Triatobatrachus, which is from the early Triassic of Madagascar. And this is an impression fossil. And from this impression, we can tell that this is a transition fossil, where it has some frog-like characters, particularly in the head, but then also has many salamander-like characters, including an elongated vertebral column and a pelvic-like girdle, or a salamander-like pelvic girdle. And then there's a gap in the fossil record to the early Jurassic, where we have Procellarius, which is thought to be the earliest representative of a jumping frog, where it has a pelvic girdle that looks more like a modern frog, including a Euro style and an elongated ilia. And then when we get to the mid-Jurassic, there are a few additional fossils of fossil frogs that basically have a modern frog skeleton, and these are sometimes even placed within the crown group. But in general, Triassic and early Jurassic frog material is very rare, and we have many of these gaps that persist. And so a few years ago, my collaborators and I actually described the first stem Silentian fossil from the late Triassic, but it's just an isolated single ilium that is elongated and appears to be more similar to living species of frogs than even Procelarius. So from this single microfossil, we think it may be possible that the highly derived body plan of frogs even evolved earlier into the late Triassic over 200 million years ago. So frogs have had this highly derived body plan for a very long time. But I really do think more microfossil work is needed to better understand the early evolution of frogs. But taken from what we know about living species and also from the fossil record, this has led biologists to conclude that frogs are highly morphologically conserved, despite that they are found throughout the world and in virtually all non-marine habitats. Or as Bob Inger once put it, a frog is a frog is a frog. They're really unmistakable as a group. But one aspect of their anatomy that has been understudied using contemporary methods is the head, which is what I studied for most of my dissertation at the University of Florida. And the vertebrate head is obviously a really important structure and a really integrative system because it's essential for housing or for sensory functions and housing the brain. It's critical for the acquisition and processing of food. And it's also really important in communication, competition, and defense. So all of these various functions result in trade-offs that result in this really integrative system. There are also many constraints that influence the vertebrate head, including architectural constraints such as allometry. So we know that vertebrate head morphology differs from looking at very small taxa versus very large taxa. And then, of course, we also have the influence of evolutionary history or phylogenetic signal, where we see species that are more closely related to one another have a more similar head shape or head anatomy overall. And so today I'll be talking about a few different aspects of the head morphology in frogs, including overall skull shape, the evolution of hyperossification or increased mineralization, and the dental diversity or tooth diversity in frogs. And much of my research uses computed tomography or micro CT scanning to study the internal anatomy of amphibians. So this is the CT scanner that we have at the University of Florida. And we've been using this machine a lot over the last few years to CT scan thousands of vertebrates for the Open Vertebrate NSF grant. And the goal of this grant is actually to CT scan every living genus of vertebrate and make it openly available online using the MorphoSource platform. And so all of the scans that I'll be showing today from amphibians were completed through this grant, and they are now publicly available online for downloading and use for research. So today we're going to start off by talking about hyperossification. During development, the skull begins as a largely cartilaginous structure that undergoes mineralization and is replaced with bone. But the degree of mineralization can really vary across species, and frogs are a great example of this. So some species end up having an underossified skull as an adult, as you can see here, where they can even have an unossified skull roof leading to an opening or a fontanelle on the top of the skull. While other species have a hyperossified skull where extramembrane bone is laid down that results in ornamentation. And so you can see the ornamentation on the skull here, which includes ridges, crests, and pits and grooves. And hyperossification is also associated often with the increase in size and fusion of elements. And this trait is not limited to frogs. It's actually a reoccurring feature in vertebrates. And we know it's evolved several times in bony fishes. All living species of crocodilians have hyperossified skulls today. Several different groups of lizards have evolved this trait. And it was also a reoccurring feature during the early evolution of tetrapods, such as you can see here in this temnospondyle. But this trait of hyperossification has been not very well studied for its evolution and function in any of these groups, but particularly in frogs, there has not been much work done on this trait. So I had a number of questions I wanted to answer related to hyperossification in frogs, including to test if it is linked to a particular head shape or head size. And to test this, we generated a large sampling of frog skulls of 158 species using CT scanning. And this is representing all 54 described living families of frogs. And then I used 3D geometric morphometrics to quantify that skull shape variation. I also wanted to test how many times hyperossification has evolved across all living frogs. And to do this, I used Bayesian ancestral state reconstructions to estimate the number of origins using an existing supertree for amphibians. And then lastly, I wanted to test for possible functional associations with hyperossifications. And a number of hypotheses exist in the literature that were largely developed by Linda Trube at University of Kansas and her students. And these include that hyperossification may be linked to preventing evaporative water loss. So all frogs have highly permeable skin for respiration that can lead to high rates of water loss. And so I hypothesized that this trait may be found in frogs that occupy certain microhabitats that are usually associated with drier environments, such as fossorial habits. It's also been suggested that hyperossification may be linked to strengthening the skull for biting. So you can imagine having a more robust skull with increased mineralization could help facilitate larger bite forces in these species. And so we wanted to see if there was an association between hyperossification and diet of frogs. Most frogs are considered gape-limited predators. Well, they'll eat anything they can fit in their mouths, but most frogs are very small, and so they usually will only eat invertebrates. But some species are capable of eating larger prey items, including other vertebrates. So we specifically wanted to test if hyperossification and vertebrate predation in frogs are linked. And then lastly, it's been suggested that hyperossification may be linked to protecting the skull or protecting the head against predators. And there are certain behaviors that frogs have, such as Phragmotic defense behavior, that has been suggested to be tied to the origin of hyperossification. So to test these various hypotheses, we collected trait data from the literature and then ran phylogenetic Mankovas to test for functional associations between hyperossification, skull shape, and these traits. So the first part of this project was generating all that CT data for frog skulls. And here we have a small sampling of frog skull diversity. And you can see there's like a pretty amazing amount of diversity in frog skull shape variation. And once we had this data, I was able to use 3D geometric morphometrics to visualize and quantify this variation in plots like these. So this is a morphospace plot where each point in this plot is a single species skull. And the points that are close to one another are skulls that are similar in skull shape. And then points that are very far away from one another in this plot are very different in skull shape. And the lines interconnecting these points is actually the overlaid phylogeny. So this is a phylomorphospace. And we can begin looking at these plots to get a better understanding of the anatomical differences that are driving the variations seen across frog skulls. So here we have the three first principal component axes of variation describing most of the variation across all living families of frogs. So this first PC1 axis of variation is largely describing the differences in the relative jaw joint position. So species with negative PC1 scores have an anteriorly shifted jaw joint or a narrow mouth, and species with positive PC2 scores have a posteriorly shifted jaw joint or a very wide mouth opening or a wide gape. The second axis of variation is largely describing how flattened or how tall the skull is. And then the third axis of variation here is describing how narrow the skull roof is. So negative PC3 species have a very narrow skull roof and a large opening for the orbits, while positive PC3 species have a very wide skull roof and a narrow opening for the eyes. And you'll also notice that there is different shapes in these plots. So circles represent species that lack hyperossification, and squares represent species that are hyperossified. And you can see that all hyperossified taxa are basically separated and are below this red line in negative PC2 space. And when we ran a phylogenetic MANOVA, we found that in general, hyperossified species have a difference in mean head shape than non-hyperossified species. So it appears that this trait does have an influence on skull morphology. And we can use these plots to look at differences in head shape and hyperossification in the context of microhabitat use. So now we have the same plot, but with the points colored for whether each species is aquatic, arboreal, fossorial, and terrestrial. And we found that all microhabitat categories significantly differ from one another in mean skull shape. And this is most evident by looking at the distribution of fossorial and aquatic frogs, where they're almost completely non-overlapping. So the brown points here versus the blue points here. But we found that hyperossified frogs are present in all microhabitat categories, suggesting that this trait is not limited to arid environments. And in fact, we found that some of the most fossorial frogs that live in some of the most arid places in the world lack hyperossified skulls, but then there are several fully aquatic species that have hyperossification. But we can now look at how skull shape is generally associated with microhabitats in a few different species. So here we have 3 highly fossorial frogs that are found on 3 different continents and are found in 3 different families. And you can see that they all have a pretty similar skull shape, including a pointed snout, a anteriorly shifted jaw joint or a narrow mouth, and a very tall skull. And so these species have convergently evolved on this skull shape. And these are also all 3 species that are forward burrowing frogs. So it isn't surprising that they have this derived head morphology to aid in their locomotion style. And then on the right here, we have 3 highly aquatic frogs that are again in 3 different families and are found on 3 different continents, but they still share similar skull characteristics, including a compressed skull or a flattened skull and a really wide opening for the orbits or a really narrow skull roof. And so again, these 3 species have convergently evolved these skull traits, and they share these microhabitat characteristics. Next, we're going to look at feeding biology. And so as I already mentioned, most frogs are invertebrate predators, but a few species are known to be vertebrate predators. And so we look through the literature to find every species that has a record of consuming other vertebrates. And then we were able to demonstrate that vertebrate and invertebrate predators significantly differ from one another in mean skull shape. And we can see that in this plot now, where basically all of the vertebrate predator frogs are colored in orange, and they're all in positive PC1 space, which is associated with that posteriorly shifted jaw joint or having a large gape, which would be useful for eating large prey items. And we also found an interesting interaction effect between hyperossification, diet, and skull shape. And you can see that all of the hyperossified vertebrate predators, so now this is now the orange square points, all fall out in this novel region of morphospace here that is not occupied by any other taxa. And these points are representative of several highly derived frogs that are distantly related from one another. And so they have converged into this region of morphospace. And many of these species share other traits in addition to their extreme head morphology, such as the presence of odontoid fangs represented by these horizontal bars. So most frogs lack teeth on the lower jaw. Their lower jaw is edentulous, but some of these species have these bony fangs that have evolved that are thought to function like teeth in that they aid in being able to inflict a bite-like wound. And so these hyperossified vertebrate predators have also seemingly convergently evolved this trait in order to consume large prey items. And so here's a sampling of our hyperossified vertebrate predator frogs that are again in 6 different families and are really found throughout the world. And again, you can see that they have very similar characteristics in their skull morphology, including that posteriorly shifted jaw joint, a relatively tall skull, and they also have some novel connections that we don't see in most frog species, such as the articulation between the zygomatic ramus of the squamosal and the maxilla bone. And so this articulation likely aids in having larger adductor musculature that would facilitate larger bite forces. And you can also see some external similarities across these frogs, such as having these eyelashes. And it's thought that these eyelashes may be present in frogs that are particularly specialized to be sit and wait predators that would be feeding on larger prey items. Lastly, we're going to be looking at phragmotic defense behavior. So this is a trait that's often thought to be associated with invertebrates, actually. It's basically the behavior of using any part of your body to block or plug the entrance to burrows and tunnels. And so this is an example in a phragmotic ant that has a really expanded head to block the entrance to its burrows. And this trait is also found in beetles and some spiders where they have expanded abdomens to block the entrance to their tunnels. It turns out some frogs are also described to be phragmotic, where they use their heads to block tree holes, rock crevices, or in the case of this picture, bromeliad axles. And so we went through the literature and identified all known species that have phragmotic behavior. And in our analyses, we identified that they all share a very novel head shape in this region of morphospace here that is, again, not occupied by any other frogs. And it turns out that phragmotic frogs have evolved this novel skull shape at least 4 times independently. And all phragmotic species have a hyperossified skull. So it seems like there's certainly a tight correlation between this skull morphology and this behavior. And historically, it was thought that phragmotic frogs do this behavior to prevent evaporative water loss because they're often found in pretty arid environments. But more recently, it's been suggested that it might actually play an important role in predator defense because there's some evidence that the hyperossified skull that has spines coming off of it might aid in a venom delivery system. So it was shown in 2015 that there are large poison glands near these spines of the skull that may facilitate a transfer of venom to a potential predator if a predator hits up against this frog skull. And so the spine of the frog skull would actually pierce through the skin through these venom glands and then into the predator, which is pretty amazing. And here there's a sampling of the phragmotic frog species that are known. They're really weird frogs, even on the outside. They have really bizarre head shapes to facilitate this phragmotic behavior. And you can see they also have a really bizarre skull morphology that is really characterized by having a really flattened skull and then also a really expanded skull roof. And it's likely that this expanded skull roof also helps protect the eyes of these frogs when they're undergoing this phragmotic behavior. And overall, we found that hyperossification has originated over 30 times in living frogs, and that can be seen in this phylogeny here by all of the black points around the edge of the phylogeny. Those are independent origins of hyperossification. And we found that it has evolved in phylogenetically and ecologically diverse lineages. We found that skull shape overall is correlated with microhabitat use and size in frogs, but these two traits are not tightly linked to hyperossification. But we did find that hyperossification is repeatedly linked to the expansion of the head into multiple novel shapes and functions, particularly in carnivorous frogs that are eating other vertebrates and those phragmotic frogs. But I do think there's more work that can be done on the possible functions of hyperossification, particularly related to osmoregulation. And I also think more work could be done on hyperossified species in other vertebrate groups to see if we see similar patterns related to these traits. And if we look to the fossil record, it turns out that hyperossification is actually pretty common in Anuran fossils, including some of the most iconic fossil frogs known, such as Bialzo from the late Cretaceous of Madagascar, currently thought to be the largest frog that ever lived. as well as this the Mastosaurus, which is an amazing mummy fossil from the Eocene of France that was all recently CT scanned and showed to have basically a perfect skeleton underneath the mummy. And our framework, I think, will be useful in paleobiological studies of fossil frogs, especially when the 3D structure of the skull is recoverable, such as these taxa shown here. And just as an example, these taxa shown here are not only just hyperossified, but they also have particularly wide skulls with the posteriorly shifted jaw joint and a bony articulation between the zygomatic ramus of the squamosal and the maxilla, suggesting that these frogs may have specialized on eating large prey like other vertebrates. And this has in fact been suggested for some of these, such as Beelzebufo, as depicted here, eating a small theropod dinosaur. But identifying the placement of these fossil taxa into the frog tree of life has remained challenging because it seems that hyperossification results in artificial groupings based on homoplastic characters. And this was shown really well by Bays and Gomez 2018. So all species in this blue box here that artificially form this clade are hyperossified, and this includes fossil taxa and living species. But we know based on molecular phylogenetics that the hyperossified living species in this clade are actually very distantly related from one another. But they had several recommendations to alleviate this and demonstrate it as well, including generating a broad taxonomic sample for these phylogenies, using a molecular scaffold tree, conducting sensitivity analyses, and even downweighting characters possibly linked to hyperossification because of the homoplasia it seems to generate. But I think of all of these approaches, maybe what might help us best is really increasing our taxonomic sampling of living species, because there is so much diversity in living taxa that is often underutilized. And now that all of these skeletal CT scans are available on Morphosaurus, I think that will be, a real possibility for future studies. And hyperossified fragments are also really common in the microfossil collections throughout the world. So here we have a sampling from India to North America to Southern Africa. And all of these hyperossified frog microfossil fragments are also really challenging to identify to their taxonomic position. For most of the cases of these fossils here, it's unclear if these fossils even belong to living frog families or not. But I do think using broader comparisons with living species and possibly focusing on characters not related to hyperossification may help us identify the taxonomic placement of these fossils. So hyperossified maxillae are relatively common in the fossil record, and I wonder if there's any taxonomic traits we could gain from better studying the microanatomy of teeth, for example. And with that, I'm actually going to shift gears here and start talking about the evolution of vertebrate teeth. So vertebrate teeth originated over 400 million years ago, and it is thought that these structures facilitated the adaptive radiation of jawed vertebrates during their transition to active predators. And teeth are maintained today in most vertebrate clades due to their importance in the acquisition and processing of food. But complete tooth loss or edentulation has evolved at least 8 times in living fishes and amniotes, and it often seems to be associated with the evolution of a secondary feeding tool, such as a beak, baleen, or a specialized tongue. Interestingly, some groups have seemingly never lost teeth, such as cartilaginous fishes, which not only have teeth in their mouths, but are also covered with teeth on their body because their skin are placoid scales, which are effectively teeth. And then there's also no evidence of complete tooth loss ever occurring within squamates. But we know that complete tooth loss has occurred at least 3 times in bony fishes, including in seahorses and pipefish. We also know teeth of this has been lost at least 3 times in living mammals, in baleen whales, anteaters, and pangolins. And then teeth were lost in the most recent common ancestor of turtles, and it was also lost in the most recent common ancestor of living birds. But one group that has been really understudied for dental diversity is amphibians. We have 3 living orders of amphibians today. We have the salamanders, which are generally thought to have teeth, the caecilians, which is an order of amphibians only found in the tropics, and they're entirely limbless and superficially may look like giant worms, but they are vertebrates and amphibians. They also generally are thought to have teeth. And then the third order, of course, is frogs. And some groups of frogs are known to lack teeth, like the true toads in the family Bufonidae, but it's unclear across all anurans if there are many instances of tooth loss or not. And so I was interested in better understanding the dental diversity of amphibians, starting off with asking some simple questions such as how many times have teeth been lost across amphibians, and is tooth loss correlated with diet or changes in body size, as has been suggested in some of the other vertebrates. And to test these questions, we again relied on CT scanning, but really increased our sampling to over 500 species and over 500 amphibian genera. So at this point, this data set is almost every known species or almost every known genus of living amphibian. And then we again use Bayesian ancestor state reconstructions to estimate the evolutionary history of teeth. And we compiled diet data, again, from the literature for phylogenetic correlations. And so here we have the skull of a typical frog showing what their dentition often looks like, where we have teeth on the premaxilla and maxilla bone of the upper jaw and teeth on the vomer, which is a bone on the palate. And as I've already mentioned, nearly all frogs lack teeth on the lower jaw, on the dentary bone here. But as I've already and it's thought that the loss of teeth on the dentary bone may be associated with the highly specialized tongue that most frogs have that they use often for feeding. But as I've already mentioned, some frogs are known to have completely lost teeth. So here's an example of those four elements all being completely toothless. When we move into salamanders, they're generally, again, thought to have teeth on the upper jaw, lower jaw, and palate. And this is actually a skull of Aeneades lugubris, a salamander found in the west coast of North America. And they actually have quite large teeth. And then here is a CT scan of a caecilian, again showing teeth on the upper jaw, lower jaw, and palate. And again, caecilians can have frighteningly large teeth for their body size. And in general, salamanders and caecilians have larger teeth than frogs. The teeth of frogs are almost always very small, usually less than 1 millimeter long, which may be why they've been understudied. Jumping right into the results, we confirmed that all genera of caecilians and salamanders maintain teeth, but we found that over 130 frog genera across 19 families are entirely edentulous. And our ancestor state reconstructions suggests that complete tooth loss has evolved over 20 times in frogs. So this is a much higher occurrence of edentulism than any other vertebrate group. We also found several possible reversals from edentulous to toothed jaws, but these cases really require further investigation. But I will highlight one case study of a dental reversal later in this talk. We also wanted to test if tooth loss is an ordered process by comparing discrete character evolution models. And we really wanted to test if vomerine tooth loss or tooth loss on the palate precedes complete edentulism in frogs. And so we wanted to test this hypothesis because when we were going through all these CT scans, we noticed that it was quite common to see teeth maintained on the upper jaw, but the absence of teeth on the palate. So we thought perhaps this was an intermediate stage of lineages on their way to complete tooth loss. But we found that the number of transitions to edentulism is similar between fully toothed jaws and palates, leading to complete edentulism is very similar number as the intermediate with tooth loss occurring on the vomer. So it does not seem like complete tooth loss is ordered process, and there's more than one way that frog lineages have become completely toothless. Next, we were interested in looking at that correlation between complete tooth loss and diet. And for these analyses, we restricted our data set to just frogs, and we compiled frog diet data for about 260 genera of frogs, representing almost every living family. But through this compilation of data, we realized that there are actually well over 100 genera of frogs that have no published diet records whatsoever. So there are a lot of living neuron lineages where we are just lacking basic natural history data still. But once we got all that diet data, we then searched for records of frogs that specialize on eating ants, termites, and mites, a diet specialization called microphagy. So there are some species of frog, such as this frog here from Southern Africa, that eats almost exclusively these invertebrate prey. And then as I've already mentioned, most other species are considered generalist predators, where they eat a broad diversity of invertebrates and sometimes vertebrates. And then we conducted phylogenetic correlations in base traits, comparing independent and dependent models of character evolution, and found really strong evidence for correlated evolution between microphagy and edentialism. And you can see that on this plot here, where red branches represent edentialist lineages and black circles represent that microphagy diet specialization. And you can see these two traits are coupled in several places throughout the phylogeny, suggesting that they have repeatedly co-evolved in frogs. And there are several interesting groups of frogs that lack teeth and are microphagous. And this includes several convergent groups that are brightly colored and are known to sequester alkaloids from their prey to become toxic themselves. And interestingly, most of the prey they eat to get these alkaloids are ants and mites. And so this includes two separate groups of South American poison frogs, the Madagascar mantelids, myobatrachid toads from Australia, and the melanophreniskis toads from South America. We also identified several lineages of highly fossorial frogs that have both edentialism and microphagy, and most of these species spend nearly their entire life underground, and this includes while they're feeding. So again, they're eating almost exclusively ants and termites, and it's actually been shown that several of these species will sometimes actually live within the tunnels and burrows of termite colonies, for example. So there seems to be a strong link here between the habits of these frogs, their diet, and their skull anatomy. It's important to note that it's not a perfect correlation, however. We do have a few examples of frogs that specialize on eating these invertebrates, but still maintaining teeth. And this is true for the Sphenorhynchus hatchet frogs of South America. They can still have teeth, although they're incredibly small, and they eat up to 90% of their diet is of ants. And then we also have a few species, such as this pippa frog from South America, that is entirely edentialist, but eats a broad diversity of prey. This is a really weird frog, though, that is not only toothless, but also tongueless. And it is a fully aquatic frog that feeds almost exclusively by suction feeding. And because of this derived feeding mode, it can consume really large prey, including fish. But overall, we found that there is rampant tooth loss in frogs with over 20 independent losses of teeth. And I'd argue that we really can't understand dental diversity in vertebrates or tooth loss with ignoring amphibians, because I think, I'd argue that tooth loss in vertebrates is largely a story about frogs, at least when considering living taxa. We also found that association between tooth loss and the diet of ants, termites, and mites. And this is an interesting correlation because it's also been shown in other vertebrate groups, including mammals. And even in squamates, there are a few taxa that still have teeth, but they're greatly reduced. And they also specialize on eating ants and termites. And so this includes the blind snakes and then a group of strange legless geckos from Australia that are in the genus Aprasia. We found no support for a reduction of body size promoting tooth loss. So this histogram here is a distribution of frog body sizes, and each species is colored red for edentialist or blue for toothed, and you can see that the distribution of body size is almost entirely overlapping. But we did find that edentulous frogs have a narrower, lower, or shorter jaws than tooth species, which again makes sense because if they're almost exclusively eating very small prey items, they do not need a wide gape. Through this study, we were able to demonstrate that edentulism has occurred in both the smallest and largest known vertebrate species. So that paedophrydian frog from the beginning of my talk is entirely edentulous. And then, of course, the blue whale is also edentulous. So it seems like tooth loss is not associated with any particular body size across the entire spectrum of vertebrates. And some of the postdoc work that I'm starting up right now at the University of Florida and Harvard is focusing on the developmental genetics of frog teeth, which has really never been studied at all, and in the hopes that I will be able to identify some of the molecular mechanisms of convergent tooth loss. Because through this study, we've demonstrated that tooth loss in frogs is really an unparalleled opportunity to begin asking questions about the molecular mechanisms of convergent phenotype evolution. Now I'll quickly highlight one example of a dental reversal focused on the re-evolution of mandibular teeth in frogs. And so I've already mentioned that lower jaw teeth are always present in salamanders and caecilians, and frogs lack teeth on the lower jaw. And this is including even all of the proto-frogs described, and including Triodobatrachus already seem to have lacked teeth on the lower jaw. But there is one species of frog deeply embedded within the frog tree of life that has mandibular teeth, and that is Gastrotheca guntheri. And this species has been thought to have mandibular teeth since it was described by Boulanger in 1882. But surprisingly, no work has ever been done to investigate the microanatomy or development of these structures. And so I suspected that this might actually be a case where we have odontoid fangs or pseudoteeth on the lower jaw of this frog, and it was simply never closely investigated. And this species is very rare, even in museum collections, and is possibly extinct today. It hasn't been seen since 1990 in the cloud forests of Ecuador and Colombia. It calls home. But we were able to get a few specimens to do micro CT scanning and histology. And we were in fact able to confirm that this single frog does indeed have true teeth on the lower jaw. So here's a comparison using CT scanning of the upper jaw teeth versus the lower jaw teeth. And you can see they're almost identical in being bicuspid in shape and being pedicellate teeth. And then when we looked at histology, we were again able to confirm the presence of dentin, enamel, a pulp cavity in these lower jaw teeth, and even the cells that produce these mineralized structures, such as a melioblast and odontoblast. We also found the presence of the dental lamina, which is this stream of tissue right here, which gives rise to tooth buds, and it plays an important role in the replacement or regeneration of teeth. And we compared this anatomy to closely related species, and again found that the lower jaw teeth of Gastrotheca guntheri are nearly identical to the upper jaw teeth in closely related species. And we also looked at the lower jaw in these closely related species, and not surprisingly, found no evidence of any of these structures. The mechanism of how this single frog species regained teeth on the lower jaw remains unknown, but I suspect there may be a suppressed tooth development program in the lower jaw of all frogs that gave a mechanism for guntheri to regain these true teeth. And this is something I'm currently investigating too for my postdoc. I also now wonder if there are any amphibian dentaries with teeth in fossil collections currently assigned to salamanders that may actually be from frogs, because if a single species was able to regain teeth, I don't see why other extinct taxa could have as well. And when we look to the fossil record again for edentulous fossil amphibians, there is no evidence of complete edentulism, as far as I'm aware, in any stem tetrapod or Albinerptontids, the fourth putative order of list amphibian, or in any fossil salamander, caecilian, or protofrog. So it seems like complete edentulism first evolved within amphibians and crown group frogs. And there are several edentulous fossil frogs that have been described. Most have been within the Pipoidea, which is made up of the families Pipidae and Rhinophrynidae. And actually, most have been assigned to the family Pipidae, which is that tongueless group of frogs. And there are 3 genera within Pipidae today that lack teeth completely. That Pippa species has 3 species that lack teeth. And then there are also 2 genera in Africa, Hymenocyrus and Pseudohymenocyrus, that are also completely edentulous. And those are closely related to Xenopus, the African clawed frog, which actually still has teeth today. But all of these living edentulous pipids are aquatic suction feeders. And based on the anatomy of the fossil pipids that have been described, these were also likely aquatic suction feeders. And we also have one edentulous rhinophrynid fossil from the Eocene of Wyoming. And there's one living species of rhinophrynid, the Mexican burrowing toad, which is a highly fossorial frog and a known termite specialist. And the anatomy of this fossil taxon is very similar to Rhinophrynus. And so it has been suggested that this was likely also a fossorial termite specialist from the Eocene. And if we look at the phylogeny of pipids specifically, including these fossil taxa based on recent reconstructions, we have on the upwards of 6 independent losses of teeth within pipid frogs alone. And so this just reiterates that teeth in frogs seem incredibly labile. And there are also 2 non-pipoid fossil frogs that both actually have a connection with Alberta. The first is Theatonius, described by Richard Fox, and the second is Tyrrhiobatrachus, described by Jim. And it's likely that these represent 2 independent cases of tooth loss in non-pipoid frogs during the late Cretaceous. And because these frogs are likely not pipids, they're also likely not aquatic suction feeders. And based on the findings that we have from our extant frogs, I think it's perhaps possible that these fossils represent actually the earliest cases of microphagy in frogs. Just speculation, but I think it could be an interesting hypothesis. And overall, I hope as we gain a deeper understanding of the biology of living anurians, we'll be able to start breathing some life into these fossil frogs. And with that, I have several people to thank, including all my co-authors for these various studies, the many institutions, curators, and collection managers that loaned us specimens. We now have CT data from specimens from well over 20 collections from throughout the world. The vast majority of this work was done when I was a PhD student in the Division of Herpetology at the Florida Museum. And I would also like to thank all my funders, including NSF. And with that, I would be happy to take any questions.
All right, Dan, thanks a lot. That was really interesting and certainly helped distill down a lot of your work. And I appreciate that because it's been a while since I've read your papers. So we do have a few questions. And I'm going to start off with a few of mine. I'm going to start with an easy one first. I was really struck in some of the sit and wait predatory frogs, the what you call the eyelashes. Mm-hmm. And I'm wondering if you have any thoughts on why they would have those kind of structures.
It's a good question, and I really don't know, but my best guess is that it may function to camouflage the frogs more and really break up their appearance while sitting in. Those frogs are often sit and wait predators in leaf litter habitats, so it may function for mimicking leaf-like structures and just breaking up their body plan is my best guess.
Right. Yeah, that makes sense. All right. I was also interested, so talking about hyperossification. I was struck by the fact that living pelobatids, so pelobades, have fairly hyperossified skulls. Yet, to my knowledge, I'm not aware that they're either eating particularly big prey or that they're phragomatic. Mm-hmm. Can you comment on why they might have the sorts of skulls they do?
That is a great point. And it's true that scapiopids are an excellent example where they're outliers in my data set and don't fit either explanation for what we currently have. And that is, in fact, why I do think, investigating the potential influence of osmoregulation related to hyperossification in certain cases might be important, because a lot of paleobatids can occur in drier habitats. And so I do think there may be a link there. But yes, I'm interested in hyperossification in paleobatids specifically because they don't seem to fit the trends for the rest of my data set.
Right. Yeah. That's not unusual in biology.
Absolutely not.
Those exceptions can be annoying, but often they're the most interesting.
Absolutely.
All right. So my colleague Caleb Brown here at the museum is interested in knowing, does hyperossification in frogs increase the chance of fossilization? And if so, how might one deal with potential bias in our sampling of frog fossils?
Yeah, that's something I've thought about a bit. And I do think hyperossified fossil frogs likely do fossilize better, which may be why they are so common in the fossil record. That being said, I don't actually have much microfossil experience, so it would be interesting to look just at the ratio of hyperossified fragments versus non-hyperossified fragments. And even just to see, if that ratio changes through time or if it's a kind of constant ratio. But as far as dealing with that bias, if one exists, yeah, it's a great question. And I'm not sure what the best approach would be to deal with that bias.
Yeah, well, I can tell you in the collections I've worked with that it's actually pretty rare to find a non-hyperossified frog.
Really?
Wow. Great kind. It's actually kind of exciting when you do.
Yeah.
So we know they were out there, but yeah, I imagine there's quite a bias towards the preservation of hyperossified frog bones.
Yes, and of course I biased on that, but the majority of frogs living today are certainly non-hyperossified.
Right. Yeah. Yeah. Okay. Question from Johan, who was interested in what sort of landmarks you were using for your analyses.
Yeah, so we did use fixed landmarks, and it wasn't. We did not use a ton of landmarks like some of the studies are using today. We had 36 fixed points across the skull of frogs, and we had to be. We were really limited in the number of points we could use in the end because we wanted to make sure we were only using homologous repeatable points. And when we were looking across, all living families of frogs, there's actually a lot of variation there. And so that kind of limited us to that number, but they were largely associated with basically the articulation meeting points of certain bones and the edges of bones to make sure that we were getting homologous points.
All right. And that will, of course, all be in your papers. So.
Yep, absolutely.
Absolutely. Okay, question here about Belzebuthos. And I just want, as an aside, thank you for showing one of those reconstructions of it gobbling down a little dinosaur. I always enjoy those sorts of images. Sender is asking, does Belzebuthos share the same wide gape and position of the skull jaw articulation and the same position in your polymorph space with other modern predatory frogs?
Right. So based on the fossil material that we have of Belzebuthos, it definitely seems like it has a very wide gape and is very similar in skull morphology to ceratophrygid fossils like the Pac-Man frog, although it's unclear if it's actually closely allied to ceratophrygid or not. But we did not directly include Belzebuthos in our analyses because it's incomplete. We would have had a lot of missing landmark data. But I suspect that if we did add it to our data set, yes, it would fall out within that region of the hyperossified vertebrate frogs, and may in fact even be the most extreme case in our data set if we were to include it.
Right. So that leads me to ask, do you have plans down the road to try and include some of those hyperossified fossil frogs into that sort of analysis?
Yeah, I would love to at some point, yeah, include a bunch of the fossilized frog taxa that have the three-dimensional conservation that would allow us to do it. Yeah, it's definitely something I've thought about doing.
Right, yeah. Put it on your to-do list.
Yeah.
With dozens of other projects. Another question here. So hyperossification pressures or explanations that you see in living frogs, can those be extended backwards to other amphibians, other early tetrapods?
Yeah, I think it's possible. Interestingly, when you get into the hyperossification literature of some other vertebrate groups, there's often a strong focus on thermoregulation, like within crocodilians. I do think it's been suggested for temnospondyls, for example, that it might play a role in reinforcing the skull for bite force purposes. I can't remember if there's been ever a suggestion of temnospondyl hyperossification being related to osmoregulation or thermoregulation, but I definitely think similar analyses could be done with some of the fossil taxa to see if we see similar patterns or not. Yeah.
Yeah, there has been some suggestions for osmotic regulation and as some sort of mineral or ion sink,
right,
for them, that they could draw on that in times of stress.
I think it's even been suggested in lizards, for example, to be a storage of calcium for like geckos, for example. So there seems to be several possibilities across all of these groups.
Right. Yeah, there's unlikely to be a one explanation fits all.
Yeah, absolutely.
So Amy Henrici, my colleague at the Carnegie, is asking are there examples of non-hyperossified fossil frogs preserved in lake deposits?
I think she might be suggesting that there are, from like a lot of the pipid fossils that exist are non-hyperossified for sure. So I think that might be part of the biasing, is we do see non-hyperossified frog fossils in areas where they're more likely to be fossilized. I think that's, yeah.
Yeah, there definitely is that. But there's also some nicely hyperossified ones in lake deposits as well, right?
Right. Yeah. It's interesting.
Yeah. Unfortunately, a lot of the microvertebrate fossil localities that I work are not lacustrine deposits, so they're more river and floodplain ones. So
right.
Yeah. So I have a question for you about the one species of Gastrophica that has teeth on its lower jaws. Does it still have a really good tongue that it flips out?
That is a great question. And I believe, based on, superficially just looking at it quickly, it looks like a normal frog tongue. I don't think we know any details of if it would use that tongue for catching prey, because as I mentioned, this frog was always incredibly rare. There are less than a dozen photos of this frog exists because it was always rare and now is seemingly possibly extinct. So I don't know if there's ever been a record of it being observed while feeding. So we don't know if it was still using its tongue for feeding, but yes, you could imagine having those dentary teeth really impacting its ability to flip out its tongue. So I've actually thought about, there's only a few specimens in collections, but it would be pretty fun to do one of the soft tissue CT scans to look at that tongue morphology to see if it has a modified tongue really in any way.
Right. Yeah, and it looked like they had pretty well-developed teeth on the lower jaws, not just...
Oh yeah, they are definitely well-developed. I've also wondered how that may influence the nerve innervation and blood vessel innervation of the lower jaw, because I imagine in most frogs, a lot of that has been reduced, if not lost, due to the lack of teeth. So definitely work to be done there too.
And as part of your work on looking at tooth development in frogs, are you contemplating seeing if you can tinker with the underlying genetics and artificially induce tooth formation in some edentulous frogs? As I know some work has been done like with that in chickens, for example.
Exactly. So I am currently just trying to describe the developmental genetics of frog teeth in species that are toothed, but then we will be going into toothless species and focusing on the lower jaw to see how many of those signaling molecules are still present and see if we can pinpoint any of the major ones that have been lost. And if we can do that, and we do find there are only maybe one major signaling molecule that is absent, we could absolutely try to add that back in and induce further tooth development as they've done in the chickens. That's kind of a dream, but who knows? Maybe it can happen.
Right. Yeah, well, certainly lots of stuff to keep you busy for the rest of your career, Dan.
Absolutely.
That's great. So that wraps up our questions. I'd like to thank Dan for a great talk. And I'm hopeful that one year we'll be able to bring you up here and show you some bits and pieces of frog fossils and maybe give a talk in person when we're back to doing that sort of work.
Yeah, that would be excellent. I would love to.
All right, wonderful. And next week we have a regular contributor, John Node, who's a geologist in Calgary, and he's going to be talking about the sedimentology of paleontology, how to accumulate highly fossiliferous deposits. So for those of you who are interested in that, that will be next Thursday at 11 Mountain Standard Time. So thank you very much for your attention. Hope everyone has a great rest of the week, and we'll see you next week. Bye.